At the end of interphase, the chromatid is still in its uncondensed form. The nuclear envelope is still present, and in the animal cell, the two centrosomes have two centrioles each. These centrosomes have duplicated during interphase. The chromatin was duplicated during S phase of interphase. During prophase, the chromatin condenses into identical sister chromatids, which are together called chromosomes. The nucleoli and the nucleus disappear, and the mitotic spindle begins to form. The mitotic spindle consists of the asters, which are short microtubules attached to the centrosomes, and it also consists of longer microtubules that extend outwards from the centrosomes. The centrosomes begin moving towards opposite sides of the cell. During prometaphase, the nuclear envelope fragments. The sister chromatids are still together in chromosomes. However, they begin to develop kinetochores. Kinetochores are an area where the sister chromatids are bonded together. The microtubules extend outward from the centrosomes. Some of the microtubules attach to the sister chromatids at the kinetochore. These are called kinetochore microtubules. Other microtubules overlap with other microtubules from the other side of the cell. These are non-kinetochore microtubules. In metaphase, which is the longest stage of mitosis, the centrosomes are still on opposite sides of the cell. The nuclear envelope completely separates, and the chromosomes line up along the metaphase plate, which is an invisible line along the center of the cell. In the metaphase plate, the sister chromatids are attached to the kinetochores, and microtubules from the centrosome on the opposite side of the cell is attached to each sister chromatid at the kinetochore. Non-kinetochore microtubules are still overlapping with each other. The mitotic spindle is then formed, and the mitotic spindle consists of the non-kinetochore and kinetochore microtubules. In anaphase, which is the shortest stage of mitosis, the sister chromatids separate, forming individual chromosomes. They are pulled apart by the shortening microtubules, which are attached at the kinetochores. As the microtubules shorten and recede back towards the centromeres, they bring the sister chromatids with them. This elongates the cell. At the end of anaphase, the two sides each have an equal number of genetically identical sister chromatids, as well as one centrosome and the remains of the mitotic spindle. During telophase, the nuclear envelopes of the daughter nuclei begin to form. These form from the fragments of the original nuclear envelope in the cell. At the end of telophase, the cells are left genetically identical. Each has an identical number of chromosomes, as well as nuclear envelopes and one centrosome. They also have the remains of the mitotic spindle. In cytokinesis, which is the division of the cytoplasm, the cells separate and become genetically separate cells. In animal cells, this starts while forming a cleavage furrow, which is an area of the cell that starts to split the cytoplasm. In plants, a cell plate forms, which eventually forms the cell wall. As the cells continue, the chromosomes dissolve once again into uncondensed chromatin.